Hello, microbiologists, and welcome to Chapter 7. This is Bio 235, Microbiology at Goodwin University with Professor Autieri. Good to have you all back again and into week 3. Chapter 7 that we will discuss today will focus on looking at the various aspects of elements for growth for various microbes, such as nutrition, the ecology, and the growth factors that are necessary for various microbes to acclimate and grow in certain environmental conditions. So, without further ado, we are going to hop into today's presentation. And I wanted to start today with just a little piece. We are so far into the pandemic and hopefully coming out of the tail end of the pandemic for COVID-19 that deals with much of the respiratory system. We're going to see that microbes find what we call niches or environments in the body where the optimal temperature and growth nutrients are available for pathogens to grow. We've also talked about that they require oftentimes a certain affinity or attraction for a cell receptor in various portions of the body. So you'll notice the picture here on the left. This is a microbe, gram-negative microbe, known as Pseudomonas aeruginosa, and it is oftentimes known as an opportunistic pathogen, meaning that it takes and colonizes the body when your immune system or your body is weakened due to some other primary infection. So we often see this as a case in patients that are cystic fibrosis patients and already have damaged lung tissue, permitting the microbe to get in and attach. And I just thought that was really relevant today to understand this role of the niche for different pathogens to colonize different regions of the body. So we're going to start today talking about microbial nutrition. And when we talk about nutrition, really what that means is how do we acquire the materials that are going to help us to live? And we know that humans really require two different types of nutrients. We have water, our cells could not deal without. We would fail to live without the intake of water. And then there's also the solid nutrients, things like proteins and carbohydrates that provide not only the building blocks for our bodily tissues, but also for cellular energy. So the bioelements, or the basic requirements for life, include this whole list of elements from the periodic table that you see here. Things like carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, sulfur, all the way down to some of these other nutrients that you see here, things like iron and chlorine and magnesium. The essential nutrients, when we break this down even further, essential nutrients are things that we absolutely must get from a source outside of ourselves. And this breaks down into two further subcategories: the macronutrients. And what I want you to remember about macronutrients is they are required in very large amounts. Macro means big, means many. So your macronutrients are often used for things like cell structure and metabolism, important and essential roles for a cell to grow and survive. So macronutrients end up making up components of your cell such as proteins and carbohydrates. As a rule of thumb, I want you to remember when we talk about the macronutrients, we talk about something called SPONGE. And SPONGE is an easy way, little acronym or mnemonic, to help you remember. S is for sulfur, P is for phosphorus, O is for oxygen, N is for nitrogen, C is for carbon, and H for hydrogen. SPONGE, those are the essential macronutrients that our cells need to craft some of these macromolecules, such as proteins and carbs. We also have what's known as the micronutrients, or sometimes called the trace elements. And you'll notice the word micro and trace. Those inquire that they're very small amounts. So these are nutrients that are only required in very small amounts and often support things like function of enzymes or supporting the structure of proteins. If you think back, if you've taken A&P 1 or 2, you've probably talked about the different levels of protein structure. And as we get up into the tertiary and the quaternary structures of proteins, there's a high degree of folding that takes place. And we have oftentimes these elements like manganese and zinc and nickel that get in and help to keep that folding structured so the protein is able to maintain its function or its active site. Microbes are going to get into the environment and they're going to acquire nutrients from many different places. So we're going to talk about things like the photoautotrophs in a little bit that acquire their energy from the photons of light from the sun. We're going to see that certain microbes have temperature environment requirements. We're also going to see that some microbes utilize different gases for their respiratory processes, such as carbon dioxide and oxygen. So we're going to see that there's a whole wealth of different environmental niches that microbes grow in based on the types of nutrients that they require. So we can break down our nutrients even further into what are called organic and inorganic nutrients. Organic nutrients contain carbon. That's the biggest thing whenever we talk about organic, is this presence of carbon and also hydrogen atoms. And we see this in a lot of different substances. Things like methane gas, carbohydrates, lipids, proteins, nucleic acids. These last four here, the carbs, the lipids, the proteins, and the nucleic acids, are the four essential macromolecules to human life. We also have the inorganic nutrients, and these are things that acquire a combination of other types of atoms other than carbon and hydrogen. So typically in an inorganic nutrient, you're going to find things like metals. You're going to see things like magnesium and nitrates and phosphates, gases like oxygen and carbon dioxide, but also water. So basically everything except for this carbon and hydrogen blend will be found with the inorganic nutrients. And that will be important later on as we start to talk about some of these specific nutrient requirements. When we look at the chemical content of a cell, there's a few things that kind of pop out. As I mentioned to you previously in one of our previous lectures, about 70% of a cell's contents, remember that cytoplasm, that gelatinous material, is comprised of water. Of the other 30%, about 97% of the dry weight of a cell is primarily organic compounds, with proteins being the most prevalent of all of those dry cell weight components. And we've already talked about sponge. We talked about those macronutrients. So about 96% of the cell is comprised of sponge, which makes up things like sulfur, phosphorus. This is important to know. These 6 elements are the keys to life. And again, table 7.1 in your text really paints a picture of a lot of the various essential elements and nutrients that comprise functions to a cell, that they have some significant role to cellular function. So if we break down the carbon-based life forms and we look at the 2 basic nutritional types, we get what's called a heterotroph and an autotroph. And what I want you to remember about hetero and auto, hetero means they have to get it from somewhere. Okay, they don't have it internally. They can't do it themselves. They have to get it from another source. Autotroph, when you see auto means automatic. They're doing it on their own. So a heterotroph has to get its carbon from another living thing. So anything that they get carbon-wise is going to come probably from ingesting another living organism. An autotroph, on the other hand, utilizes its own CO2 source as its carbon source. So it doesn't depend on other living things to generate its carbon supply. We also talk about factors that the organism can't create itself. So it's unable to create and synthesize some of the essential things that it needs for not only genetic but metabolic structures within the cell. And these are what we call growth factors. So growth factors cannot be synthesized and they have to be provided to the organism for survival. These include things like amino acids and vitamins. So we can classify nutrition based on two types. The carbon source, which we've talked about so far, heterotroph, which is ingested from other organisms. And we've talked about the autotrophs, which are not nutrient dependent on other organisms because they're using carbon dioxide gas as their source of carbon. We can also classify them based on their energy source. And there's two different ways to distinguish the energy source. You'll notice we have what are called chemotrophs and phototrophs. Chemo means chemical. So the energy is acquired from chemical compounds. Anytime we see photo in the biological sciences, photo refers to light. So phototrophs get their energy from the acquisition of light. And we call this process photosynthesis. Photo, where light is utilized to synthesize or create energy. And you're going to see that we're able to take these terms and merge them together to really describe not only the carbon source, but also the energy source of an organism. For instance, we can have a chemoautotroph, and what a chemoautotroph is they get their energy from chemical compounds. Their carbon source comes from CO2, that inorganic carbon form. So again, here's some examples here. This is table 7.2 from your textbook, which again diversifies the different nutritional categories of microbes. So if we take the carbon source and the energy source and we combine them together, we get our photoautotroph and our chemoautotroph. Again, energy from the photoautotroph comes from sunlight. Chemoautotroph energy comes from those inorganic compounds such as CO2. And they also provide you some examples of these different organisms. When we talk about the heterotrophs that are using organic carbon, we get things like a chemoheterotroph. Okay, chemo meaning it's going to use chemicals for its energy source. Heterotroph meaning it's using organic carbon. So humans, for instance, are chemoheterotrophs. We are converting nutrients from other organisms for our energy source. And there's two kind of breakdowns when we talk about microbes that do the chemoheterotroph. We have the saprobes, which are organisms that feed off of dead, decaying matter. We often know these as decomposers. And many times these are bacteria and fungi can fit into this saprobe group. And later on, we're going to talk about symbiotic microbes. Symbiosis is a close relationship between two different organisms. And then, of course, just like we could have a photoautotroph up here and a chemoautotroph, we can also have a photoheterotroph down here. So again, the light or energy source is coming from light, and heterotroph, again, is that organic form of carbon. So our photoautotrophs is where we're going to start. And we're going to dive in and start talking about some examples of our autotrophs. So when we're talking about photoautotrophs, we can break down the types of photosynthesis that they undergo. And we started talking about this during chapter 1. We have the oxygenic photosynthesizers, and this includes organisms such as the plants, the algae, and the cyanobacteria. And oxygenic photosynthesis means just that they produce oxygen as a byproduct of photosynthesis. So they take in carbon dioxide, They convert that to energy, and in the process, they release oxygen as a byproduct. The anoxygenic photosynthesizers, on the other hand, are organisms that go through photosynthesis, but anoxygenic. You see this, one of the tricks here to remember this is where it says ano, this no right here. Think of no as no oxygen. So they do not generate oxygen as a result of photosynthesis. So the anoxygenic photosynthesizers include things like the purple and green sulfur bacteria. I've kind of asked some of these questions in our discussion board posts to get you thinking about the purple and green sulfur bacteria. We also have the chemoautotrophs, and these are sometimes called the lithoautotrophs. And what I want you to know about the lithoautotrophs is that these completely rely on inorganic substances for their growth. So they are going to use inorganic materials for not only their carbon source, but also their energy source. And an example of this are the methanogens. And remember, the methanogens are unique because they are producing methane gas. So the methanogens grow in what are known as anaerobic conditions. And anaerobic means no oxygen. So they grow in an environment with no oxygen. However, the methanogens environment contains not only hydrogen gas, but also carbon dioxide. And they utilize those components to produce methane gas. So this is an example, again, where the methanogens are lithoautotrophs. They do not utilize any organic substances and completely rely on the presence of inorganic materials in a non-oxygen environment to produce methane. We can also talk about the chemoheterotrophs. And when we get into the heterotrophs, there are two basic categories that we want to talk about. Those being the saprobes, and we just mentioned saprobes a few minutes ago. These are organisms that feed off of dead, decaying organic matter. And a lot of times these are either opportunistic pathogens, which means they can take effect to the body when your immune system is compromised, or they can be facultative parasites. And facultative means that they can survive under different conditions. So there's multiple conditions for a facultative organism. And then obviously we know that parasites actually feed off of a host. And we said that some parasites can also be pathogens, and many are what we call obligate parasites, which means that they absolutely need a host cell for their growth and survival. We're also going to talk about how organisms move substances in and out of the cell. And we mentioned early on in our process this semester, we talked about the cell membrane, the selectively permeable barrier that ultimately decides what's able to enter and exit a cell. We're going to break our transport down into two different groups depending on the energy required or expended. We have passive transport, and one of the things about passive transport is it doesn't require any energy to facilitate the process. We also have what's called active transport, which does require the addition of energy for the process to occur. So the reason why passive transport does not require additional energy is there is a gradient that exists, concentration gradient of a substance. So in one part, we have a really high concentration, and somewhere else, there is a low concentration. And as a rule of thumb in biology, molecules tend to move in a direction from a high concentration to a low concentration. And they do this naturally, or what we call spontaneously. So we do not have to add additional energy, and we know that the cellular energy source, the cellular currency of energy, is ATP. So there is no ATP addition in passive transport. And there's three different formats of passive transport. We have diffusion, where small uncharged substances move from a region of high concentration to low concentration. These are usually things like gases or very small uncharged molecules. We have osmosis, and this is the movement of water from an area of high to low concentration. And then we can step diffusion up one more notch, and we have what's called facilitated diffusion. And this is when we need to move a substance that may either be large or possess a charge. And in order to do that, we are going to need some type of a transport protein embedded in the membrane to help facilitate or carry out that diffusion. And these are in the form of either protein carriers or channels that can move things from one side of the membrane to the other. We mentioned active transport, which now requires not only the addition of ATP for energy, but also carrier proteins, and it doesn't matter about the gradient. So we mentioned that with passive transport, things typically move from a high to a low concentration. With active transport, because of the acquisition of energy and also the carrier, we can now move things from a region of low concentration to a region of high concentration. And we talk oftentimes about these different bulk transport methods, which are considered active transport. Things like endocytosis and exocytosis. Remember, we talked about these in the viral chapter. And when we looked at animal viruses, those viruses go through endo and exocytosis. Think of EN as enter, so things are going to enter cell, cyto meaning cell. Exo, EX for exit, things are going to exit the cell. And basically what happens is they get surrounded by a piece of host cell membrane to form a vacuole. And those vacuoles then move either into or out of the cell. Pinocytosis, the way I always help students to remember this one is think of pint of liquid. Pinocytosis involves the bulk movement of liquid into or out of a cell. So we already mentioned with diffusion, we always go typically down a concentration gradient from high to low. So things are always going to want to go from where there's a lot of them to where there's less of them. And that's what you see here. If we were to take this sugar cube and drop it into this cup of coffee, the sugar cube is then going to start to dissolve, and these sugar molecules are going to move out into the liquid away from this high concentration. And that's why no matter which sip you take, if you were to stir this well, you're going to be able to taste that sugar in just about every sip of the coffee that you take, because those sugar molecules are going to diffuse out into the liquid. We mentioned osmosis, again, movement of water across the membrane. And this is when we have a difference in solutes in a solution. And a solute is a thing like a salt or sugar that causes differences in a liquid. And this diffusion or this osmosis is going to occur until an equilibrium is reached, basically until we have the same amount of substance on either side of a membrane. So I want to talk about the three different conditions that we find under osmosis. This is often what is referred to as tonicity. So we have three different tonic relationships. We have isotonic, hypotonic, and hypertonic. Isotonic is what we would call our equilibrium. It's our homeostasis in terms of human cells. We are constantly trying to get to an isotonic state. And that basically means that the water concentration in and out of our cell is equal. We have the same amount of water inside as we do outside. In order for that to occur, you'll notice that The little blue specks that you see here are solute particles. Whenever we have the same amount of solute outside the cell as we do inside the cell, there is going to be no net movement of water. It means that the same amount of water that goes out of the cell is going to be the same amount of water that moves into the cell. And you'll notice that this cell membrane is not stressed in any way. The cell is in a very content, very happy state. Now, when we get into a situation where we now start to create differences in the solute concentration both in and out of the cell, water is going to move very differently. So now, when we have a high amount of solute inside of our cell, what's going to happen is water is going to move from where it's really high to where it's really low. So if there's a lot of a salt particles inside of a cell, the water concentration is going to be very low. So water is going to want to move from the outside into the cell. And when it does this, the cell is going to swell. However, because bacteria have that strong peptidoglycan cell wall, the cell wall actually prevents the cell from bursting or popping like a balloon. So when water moves in, if a cell doesn't have a cell wall, this cell is going to continue to swell until it breaks or lyses. We also have a hypertonic solution. And that means if we were to put this cell in a solution that has a really high solute concentration outside, the water is going to move from where it's really high to where it's really low. So water in this case, since there's more solute outside the cell and less water, water is going to move out of the cell. And we're going to get the opposite effect of the hypotonic relationship, where now this cell is going to shrivel. And this shriveling, where the cell membrane actually starts to pull away from, you'll notice in this picture, the cell membrane starts to pull away from the cell wall. This is what we call plasmolysis, or the shriveling of the cell. So I already mentioned that if a cell lacks cell walls, you're going to see in the case of an isotonic, that the cell is going to be fine. The membrane is going to be okay because water is moving equally into and out of the cell. In the case of a hypotonic solution, we mentioned that the salt or solute concentration inside is much higher than the water. So since water is higher outside than it is inside, it's going to move from high to low. So water is going to go into the cell. And as water starts to pour in, this cell is going to swell and eventually lyse or break open. And in the case of a hypertonic cell without a cell wall, since the solute is higher outside the cell, okay, water is low outside the cell, water is going to move from where it's really high on the inside to where it's really low on the outside. And this cell, when it loses the water, goes through plasmolysis, where that cell membrane actually pulls away from the cell wall, and we get this shriveling of the cell. When we talk about facilitated diffusion, we mentioned again that it still goes from high to low concentration. However, now it requires a carrier protein or a channel protein to help get materials in and out of the cell. So here's an example of a carrier. Again, you'll notice here's our cell membrane. Here's the inside of our cell. Here's the outside of our cell. We have a high solute concentration on the outside, represented by these little purple diamond-shaped pieces. And we need to get them into where they're really low. But since they're large, they're not going to be able to move across this membrane freely. So they are going to bind to these specific protein carriers. And what's going to happen is, once it binds, it's going to cause a change in shape to this protein. You'll notice that here, this opening is really narrow. When this binds, what happens is this widens and allows this item to pass through into the cell. So still same thing as diffusion, where we have movement of these solid particles going into the cell from high to low. But since they're either larger or charged, they need a carrier protein to help get them across. And we've also talked about, the use of channels as well to help get things across. So we're going to talk about active transport. And again, we said the difference between passive and active transport. Active transport requires a source of ATP to help facilitate the process. Still utilizes a protein to help move things across the membrane, but now also the use of ATP as well as an energy source. So these specific membrane-bound proteins are going to attract and bind to these solutes, these items such as sodium or sugar. And once those solutes are attached to that specific site, ATP is going to activate a pump to move the material across the membrane. So you'll notice here's the inside of our cell, here's the outside of our cell. Again, we have these little solute molecules represented by the green. It is going to bind to our channel, and that substance, through the use of ATP fueling the process, is going to help transport that item across the membrane. Same kind of thing. They also have what's called translocation, and this is where you can get multiple of these substances to be able to bind and help facilitate the movement of substances across the membrane. Again, a lot of times these molecules are chemically altered or activated to be able to allow them to pass through on the membrane protein carrier. And then we also mentioned the endocytosis, which is what we know as the eating or drinking of cells. Endocytosis, as we mentioned, is, again, think of endo as enter. Things are going into the cell, and they are brought in these tiny little pieces of membrane that pinch off, and we call these vacuoles or little pockets. We can either have two examples. We have phagocytosis, which is where whole substances or cells are engulfed. And we also mentioned pinocytosis, think of pint, is where we ingest fluids into these vacuoles that move into the cell. Again, Table 7.3 does a great job of summarizing the differences of passive and active transport, so I definitely recommend taking a look at that as you prepare for exams. So without further ado, we're going to move into kind of the second phase of the chapter that looks at the environmental factors that are necessary for growth. And we talk a lot about, as I mentioned to open our presentation today, niche, which are the adaptations that organisms make in their environment. And we're going to see that there are a whole slew of different environmental factors that influence growth and survival. Things like temperature, the presence or absence of oxygen, the acidity, the presence of salt, and the presence of pressure. All influence whether organisms are able to grow in a particular niche. So we're going to start off today talking about temperature, and we're going to see that temperatures occur on a range. So we have what's known as the minimum and maximums, which are the lowest and highest temperatures that will permit growth. But we also have optimum temperature, which is where the organism will experience its highest growth rates. These are essential terms to know for exams. Oftentimes, I will give you a little scenario and ask you to tell me what type of organism we're talking about. I may say something to you like, I grew an organism in the refrigerator for 2 weeks. Then I took it out and tried to grow it on the lab bench, and it refused to grow. What type of organism is this? And really what I'm looking for is for you to pick up the fact that it only grew in the refrigerator. And in this case, we would know that as a psychrophile. These are organisms that are cold lovers. They grow at temperatures from about 0 degrees Celsius up to about 15. So extremely cold environments. We have the mesophiles. Most of the human pathogens that infect our bodies are mesophiles, and they grow right around body temperature. And then we have the thermophiles, or the heat lovers. Think thermo is heat. These grow at temperatures, really hot temperatures, greater than 45 degrees Celsius. So we're almost halfway to boiling at that point. So our thermophiles grow in these much hotter ranges, and we could even go out further to the extreme or the hyperthermophiles, which grow starting around 70 degrees all the way up to about 130 degrees Celsius. So again, our psychrophiles are cold lovers. Our mesophiles, those moderate temperatures, our human pathogens grow here. And our thermophiles are heat lovers. We can also look at the ability to grow in different oxygen environments. So we're going to talk first about our aerobes. And best trick to help you remember this is think about when you're doing aerobics. You go to the gym, you're running on the treadmill, you're on the elliptical. You are utilizing oxygen at much higher rates to continue to support the muscular energy that you are expending. So an aerobe, think aerobe, aerobics, we are utilizing oxygen. Okay, so aerobes utilize oxygen. There's 3 subcategories when we talk about aerobes. We have obligate aerobes. Obligate means it's a requirement. So obligate aerobes cannot grow if oxygen is not present. Facultative, on the other hand, as I mentioned earlier in our presentation today, facultative means that it can exist in a range of conditions. So a facultative anaerobe can grow and use oxygen, but if oxygen is absent, it can still grow. So it's kind of indifferent. It will use it if it's there, but if it's absent, it will still grow anyway. It will figure out how to utilize something else. And then we have the microaerophile, again, micro meaning tiny. Microaerophiles require a very small amount of oxygen, but won't grow at normal atmospheric levels. So these only can grow in a very small amount of oxygen, and you're not going to get growth at normal atmospheric levels. Anaerobes, on the other hand, if an aerobe utilizes oxygen, think of no, does not utilize oxygen. So these will not grow in the presence of oxygen. And we have 2 different relationships here. We have the obligate anaerobes. Again, obligate means that they absolutely have to have it. So in this case, they cannot grow for any reason in the presence of oxygen. Aerotolerant anaerobes, here's where it gets a little trickier. Aerotolerant anaerobes do not utilize oxygen, but if we were to expose it to oxygen, it will still continue to survive. It's not going to be killed. If we exposed an obligate anaerobe to oxygen, it will die. So it is possible to have organisms that are not using oxygen, but can survive and grow in its presence, and we call those aerotolerant anaerobes. We utilize a particular type of medium known as thioglycolate to help us to demonstrate the oxygen requirements of an organism. And we will utilize this in the lab during this semester. You will actually test this out and determine the oxygen requirements of a particular microbe. Based on where they grow in the tube, it's going to tell us their preference for either an oxygen-rich or an oxygen-free environment. We can also have some techniques to help us to grow our anaerobes, our organisms that cannot grow in the presence of oxygen. So most oftentimes we have things like anaerobic chambers that are filled with carbon dioxide. We have a packet that we add some different catalysts to, and it generates a carbon dioxide rich atmosphere. We also have something known as a capnophile. And capnophiles grow best at very high CO2 concentrations, higher than we would typically find in the atmosphere. So we utilize for capnophiles something known as a candle jar. And basically what that is I would take all of my petri dishes that you see here, I would put them into my candle jar chamber, and then I take just a small little votive candle. I light my candle, I put it inside my jar, and then I screw on my airtight lid. And as the candle burns and depletes all of the oxygen, it will raise the carbon dioxide levels, and eventually the candle will burn out. So now the organisms have an environment that is extremely low in oxygen and extremely high in carbon dioxide. We can also look at pH, or acidity. Organisms that are neutrophiles, which are most of our microbes, especially the ones that grow in our bodies, prefer a pH of about 7, and that's what we call our neutral pH. And these are our neutrophiles. They grow in between about a pH of 6 and 8. Acidophiles grow at extremely acidic pHs. So we're going to find these, at a pH of 1 all the way up to about a pH of 5. And then our alkalinophiles grow at basic pHs. So basically everything from 8 to 14 is where our alkalinophiles, or our basic organisms, are going to grow. We've also talked about the presence of salts and sugars that create these osmotic pressures. Certain organisms have requirements for different salt conditions. So, for instance, our osmophiles are organisms that require high salt concentrations. We also sometimes call the osmophiles halophiles, halo meaning salt. So, an obligate halophile means that it grows in high salt concentrations, places like salt lakes and ponds. We also have the osmotolerant organisms, and these do not require high concentrations of solute or salt, but they can grow if they're exposed to it. So things like the facultative halophiles are extremely resistant to salt. Things like Staphylococcus aureus, which is a common organism that you would find either on your skin or growing around the nares of your nostrils. And as we mentioned, the barophiles grow in extremely high pressures. So places where the pressure exceeds much higher atmospheric conditions. So for instance, the archaea that live at the ocean floor in the deep oceans would be organisms that are considered barophiles. With that being said, we're now going to talk about some of the ecological associations of different microbes. And we're going to talk about symbiotic and non-symbiotic relationships. A symbiosis is a close interdependent relationship between two organisms. And symbiotic relationships can be broken down into three categories. We have a mutualism, where both members in that relationship receive a benefit. We have a commensalism, where one organism receives a benefit and the other is neither harmed nor helped, so it's unaffected. And then we have a parasitism, where one organism is harmed and the other receives a benefit. So think of mutualism as a plus plus, both members are happy. Commensalism is a plus and a 0. One organism gets a benefit, the other one doesn't have anything happen to it. And then a parasitism is a plus minus. One receives a benefit, the other is harmed. The non-symbiotic relationships, these are organisms that are free living, and the relationships are not really obligatory. They're not really required for them to survive. So we have two different types. We have a syntrophy, where members are cooperating together and they share a habitat and each one is able to create nutrients that they share with each other. And we have an amensalism where some members are either inhibited or destroyed by the others in that relationship. So again, this is a flowchart from your text that compares our symbiotic relationships, such as our mutualism, commensalism, and parasitism. Remember, symbiotic relationship, they live in very close, interdependent relationships. Non-symbiotic, the organisms are free living and the relationships are not required for survival. One of the big microbial relationships that we talk about quite a bit in this course are biofilms. And these are close layers of microbes that are interacting and communicating with each other. And they form this matrice that forms and consists of organisms communicating through chemical means. And we term this quorum sensing, where they're actually sharing chemical signals to communicate between cells. So if I wanted to find a biofilm, I could go to places like, the thin film layer that forms maybe on your shower door, or if I remove the shower head and I found that buildup of that kind of murky substance, that's a biofilm. Plaque that you feel on your teeth when you first wake up in the morning, that's also a close association of bacteria that forms a biofilm. In the medical setting, we often see it on the formation on catheters or other types of surgical implants. You do not need to know the specifics of how quorum sensing works. I want you to know that quorum sensing is the use of chemical signals to communicate between cells. We also talk a lot about the interaction between microbes and humans. There are a substantial amount of symbiotic relationships that form between these two. Microbial growth, when we study microbial growth, there's a whole system that we utilize to describe it. Microbial cells divide through a process known as binary fission. And all I really want you to know is the cell makes a copy of its genetic material, its chromosome, and the one cell divides into two new cells. So we often talk about microbial growth as exponential. For one cell, it divides to two. Those two cells divide and become four. Four cells become eight. Eight cells become 16, and so on. So we get this exponential, or oftentimes called logarithmic, growth. So again, here's our example. Here's our one cell. Our cell copies its circular chromosome. Each new cell gets a copy of the chromosome, and that one cell then splits and divides into two new cells through binary meaning two, fission meaning to split. We mentioned already that the cell growth is logarithmic, so if you were to graph it, it would be on this kind of curved slope as the number of cells divides. We mentioned that one cell becomes two, those two cells become four, four become eight, and so on. So it's not a typical arithmetic growth where you go 1, 2, 3, 4, 5, but you actually get this doubling that occurs. And the time that's required for the doubling to happen is known as the generation time. So every time those two cells go through a cycle and divide, we call this a generation time. Generation times vary greatly for organisms. We have some that can do it in a few minutes. Some microbes, like Mycobacterium tuberculosis, can take several days or weeks for their generation times. If we wanted to compare population growth in a mathematical formula, we're able to do this by a particular formula that helps us to calculate population size. And this is NF stands for the total number of cells in a population. NI is the number of cells that we started with. And the 2n is basically the doubling of the number of cells in that generation, or our generation time. So if we were to input all of these different components, that would tell us how many cells around we would have in our population. So how do we study growth in the lab setting? Well, we have a few different tools that we can utilize. First of all, we have what's known as the viable plate count. And this is basically where we can take a culture and we can add a certain amount of that culture into a test tube of liquid. And then what we're doing is we're taking a little bit of the liquid out of this first tube and putting it into the second tube, taking some of that liquid out of here and into here, and so on. Some out of here into here, and right down the line. So this first tube is still going to be pretty concentrated. This tube down here is going to be very dilute. So by doing that, we're creating this dilution. And when we plate it, we're able to see, you'll notice down here, we have a very high concentration. And over here, we have a very low concentration as it dilutes out. So this can help us to determine the original population size of a culture, because we get these living growths on the plate. And what I want you to know is each colony, each one of these dots that you see, actually started as a single cell. So one cell ultimately ends up creating this mass of cells that we call a colony. The bacterial growth curve is essential for you to know. So there's 4 phases when we talk about population growth with microbial cells. Whenever we first introduce microbes to a new growth medium, they go through what's called the lag phase. And the lag phase is the portion of growth where they're getting acclimated to their new environment. Think about when you first move into a house or an apartment. The first time or the first couple of days you move in, everything's in boxes. You're just trying to get yourself in and settled and maybe get your bed assembled. You're not really having company over at that point or doing too much because you're just getting acclimated to your new environment. Once they're acclimated, they begin to grow at that exponential or logarithmic rate. And we call this the log phase or the exponential growth phase. So the bacteria are actively using nutrients, they're growing, they're dividing through binary fission. However, at some point, the nutrients that are in this tube are going to start to run out. And what's going to happen is we're going to get this plateau here that we see in stationary phase. And stationary phase is where the number of cells that are generated equal the number of cells that are dying. So there is no overall change in growth. However, as these cells start to die, you'll notice this formation at the bottom of the tube. They're going to produce toxins as the cells die. And this is now going to cause other cells to begin to die in what's called the death phase, where we have this overall gradual decline of cells in our population. So again, this slide, I'm not going to run through this again, it gives you a breakdown of our lag, log, stationary, and death phases as part of our growth curve. And we can measure this growth again in some technological ways. We have what's called the turbidometry, and this uses a tool known as a spectrophotometer. And what it does is it measures the cloudiness of our tube to figure out what the approximate growth size is. So you put your test tube into this chamber, and a light source comes on, and it shines through, and the absorbance of the light that is caught by the little machine will then tell you about how much population size is in your culture. We can also do what's known as a direct cell count. And this is basically where we put some cells on a microscope slide and we count the number of cells in the grid, and that we can use a formula to get a general count of how many cells would be in our population. We can also use what's known as a Coulter counter or a flow cytometer. And we would use the flow cytometer when it's important to count the number of cells or determine the cell size, and also the difference between the number of dead and living cells. We would want to utilize methods like flow cytometry. So, with that being said, that is the end of our presentation for Chapter 7. Feel free to swing by office hours, ask any questions that you have that you may be stuck on, and I look forward to seeing you soon. This is also the final chapter for Exam 1 of the course, so the Exam 1 materials for your preparation end at this particular point. See you all soon.